Hey friends, I'm Aarti Sharma and welcome again to my YouTube channel. In today's video tutorial, I'm gonna talk about chromatin, chromosome and chromatids. So let's get discussed. First of all, I would like to talk about chromatin. What is chromatin? Chromatin is nothing, it's simply the complex of DNA with protein molecules, right? So what is the need to DNA combine with proteins? We know that each cell contains approximately 2 meters of DNA. Now it is very important to organize this 2 meters of DNA inside the small sized nucleus of the cell. For the proper and efficient packaging of this 2-meter DNA inside the nucleus of cell, this nucleus make combination with different types of proteins in order to pack itself inside the nucleus, right? So when it associate with proteins, that structure is known as the chromatin, right? Now the structure of chromatin will vary in different phases of the different phases of the cell cycle. Here you can see that this is the cell cycle. Cell cycle basically consists of interphase and M phase. Interphase is the phase where the cell prepares itself for dividing into the daughter cell. M phase, it is the phase where the parent cell divide into the daughter cells, right? Your interphase consists of G1 phase, gap 1 phase, synthesis phase and gap 2 phase, right? So in the interphase, what happens? Your genetic material replicates, it transcribe and make lots of proteins, right? And in the M phase, which is known as the dividing cell, will divide into the daughter cells. Is it clear? So the structure of chromatin will differ in each phase of the cell cycle. If I'll talk about interphase, then we have discussed that in interphase the genetic material will replicate, it will transcribe. So it means the DNA, the parts of DNA should to be exposed to the transcription factors, to the proteins which are important for the DNA replication, right? So what happens in case of interphase? Your chromatin structure will be in a noodle like form. Right, here you can see that inside the nucleus the chromatin is visible as a noodle like structure, means the association between DNA and proteins will be weak. Okay, proteins will not make strong combinations with the DNA. Due to the weak combinations, more and more DNA will be exposed to the transcription factors as well as to the proteins, right? But if I talk about M phase, that is the dividing phase where the parent size daughter cells, in that situation the DNA is not going to replicate, nothing is going to transcribe, means no proteins are going to form. Whatever protein content is required for the dividing of the cell will be synthesized in the interphase. So what happens in the M phase? The noodle like structure of chromatin that is present in the interphase, it will begin to condense more and more. After condensation, it will take the structure of chromosome, right? These chromosomes are very important for the M phase, because M phase is the dividing phase where the parent cell will distribute equal amount of chromosome to each and every daughter cell. So for this proper segregation of chromosomes, the proper organization of DNA inside the nucleus is very, very important. Is this clear? Yes. So next is, now I hope you are clear about chromatin. This video is just to give simple clear cut idea regarding chromatin, chromosome and chromatid. Okay, so we will discuss in detail about DNA organization in our upcoming videos. Clear? So chromatin is simply when your double stranded thread of DNA combine with some proteins, it will form chromatin that looks like a noodle like structure in the interphase. But when the cell cycle approaches to the M phase, chromatin will keep on condensing, and after lots of higher level of condensation, it will achieve the structure of chromosome. Clear? So we know that chromatin is the combination complex of DNA and proteins. The major proteins which are responsible for DNA organization are the histone and non-histone proteins. The major role is contributed by the histone proteins. The amount of the amount of histone protein will be almost equal to the amount of DNA, right? The histone proteins, we have H1, H2A, H2B, H3 and H4, right? And scaffold protein, it is a which also helps in organization of DNA inside the nucleus of the cell. Here you can see that the structure of nucleosome, that is the structural unit of your chromatin. So here these ball like structure, these are the histone proteins, and this red colored thread like structure is your DNA. This DNA wrap around your histone proteins and the structure will be known as the nucleosome. So here you can see that these red colored structures are your histone proteins, and when DNA wrap around these histone proteins, they will become nucleosome, and the structure looks like a beads on string. Okay, and these beads on string in interface will look like noodle like structure, but then when they will keep on condensing, they will take the structure of chromosome, something like this. So what do you mean by chromosome? Chromosome is nothing but when your chromatin keep on condensing, after achieving the high level of condensation, it will take the structure somewhat like this. Okay, so this is the structure of your chromosome. Your chromosome, we know that we have 23 pairs of chromosome. That means we have 46 number of chromosomes, and each chromosome is present in pair. Let's say this is the chromosome number 1 and this is the another chromosome number 1. This is the pair of a chromosome, and these two structures are known as the sister chromatids. These sister chromatids are attached to each other through a central point that is known as centromere, and we have kinetochores in the centromere. Kinetochores are the structures which helps for the attachment of spindle fibers. Spindle fibers attaches on the kinetochore. Okay, and these spindle fibers help to segregate two sister chromatids during the time of cell division. Is this clear? So next is your chromatid. What is the difference between chromosome and chromatid? Chromatin is what? We know that we have 23 pairs of chromosome, right? And each pair will have two chromosomes. Clear? So the single chromosome of a pair is known as chromatid. Let's say this is the chromosome number 1, this is the another chromosome number 1. So in a pair I will call chromosome, but in a single form you can call them chromatids. Clear? So this is the difference between chromosome and chromatid. We can say that we have 23 pairs of chromosome. That means in total we have number of chromatids or 46 number of chromosomes. Clear? So in pairs you may call them chromosomes, but when you are talking about a single chromosome, when you are talking about only a single chromosome of a pair, you will call them chromatids. I hope you are clear about the chromatin, chromosomes and chromatids. I hope you understand my lecture, and thank you so much for watching my video.